पवित्रवतु ॐ चोदयेत्रे सूनृतानां चेतन्ति यज्ञं दधे सरस्वती।
Today we are going to talk about microbes as miniature industries. Under this part, we are going to learn a few basic terms related with fermentation technology and a little in details about inoculum development process. I definitely know that all of you must have heard about these substances. Each one of you here are aware of these substances. Most of these products are produced because of the activity of microorganisms. Microorganisms have been exploited for producing these useful products from the time that when microorganisms, the existence of microorganisms was not even established, nor were their characteristics known. Therefore, definitely we can say that this is a old science that we have now rediscovered again. As early as 6000 BC, the Babylonians and the Sumerians, they made wine from yeast. They produced alcohol from yeast. In fact, history reveals many such applications of useful processes wherein the microbial activity has given us a variety of useful products, mainly foods and beverages. However, the involvement of microorganisms in the production of these substances was not completely established until the studies conducted by Louis Pasteur in the later half of the 19th century. As from the standpoint of industrial microbiologist, if we talk about, we can say that the microorganisms can be considered as chemical factories in miniature. Now, this is clearly because of the fact that these microorganisms are capable of converting raw materials, which are substrates or nutrients provided to them, into a new product. Now, if this end product that is produced by this reaction of microorganisms, definitely, and if this end product has a value for human use, then it becomes an attractive option to scale up this reaction and perform it on a larger commercial scale. Two dramatic explosions that happened in the field of industrial microbiology. What were these two industrial explosions that happened? The first one was in the 1940s, which was because of the discovery of antibiotics led by penicillin, after which major industries were built up depending on the microbial activity. The second explosion came in because of the great advances in the knowledge of microbial genetics, whereby it was now possible for us to manipulate the microbial DNA and generate newer products. This technology is known as the recombinant DNA technology. And with the development of this technology, we can now do what is called as bioengineering of microbes, which can produce the products that we want them to produce for us. This particular process is definitely going to revolutionize the field of industrial microbiology. Let's now move on to learn about the major classes of microbial industrial products. These could be classified into four different categories. The first one, as we know, the microbial cells themselves could be serving as products. For example, you must have heard of single cell proteins, where these could be consumed in case of protein deficiencies. The second product being large molecules like enzymes, which are products of microorganisms. Thirdly, the primary metabolic products, and fourthly the secondary metabolic products. Now what do you understand by primary metabolic products and secondary metabolic products? If I look into the growth cycle of a microorganism, wherein the number of cells is plotted against time, most of the microorganisms will follow this particular curve, which is called as the growth curve of a microorganism. As you see, there are 4 different phases to this. This is called as the lag phase, this is the logarithmic phase, this is the stationary phase, and this is the death phase. As you see, in the lag phase there is no increase in the number of cells. Logarithmic phase, there is a rapid increase in the number of cells with time. Stationary phase, again no increase in the number of cells. And death phase, there is a decline in the number of cells. When I say primary metabolic products, these are compounds produced by the cell, by the microorganism, which is required for its own growth. And these compounds are normally and primarily produced in the logarithmic phase of growth cycle. The secondary metabolic products are also products produced by microorganisms, but these are compounds not required by them for their cell growth. Okay, and they are primarily produced during the stationary phase of the growth cycle. So this is the basic difference between the primary metabolites and the secondary metabolites. Primary metabolites produced in the log phase or the logarithmic phase, and secondary metabolites produced in the stationary phase. Primary metabolites required for cell growth, secondary metabolites not required for cell growth. So all of the major microbial products fall into these 4 categories. Okay. Let's now talk about the concept of fermentation technology. If you look at the word fermentation, and if a biochemist is told to define the term fermentation, you basically understand it as a anaerobic conversion whereby energy is produced without the participation of oxygen. The term anaerobic itself suggests that it is minus oxygen. So the term fermentation means that it's an anaerobic conversion without the participation of oxygen and making use of other inorganic electron acceptors. However, an industrial microbiologist, if he has to describe fermentation word, what the industrial microbiologist has done is, it has widened the meaning of fermentation technology. It says, or it uses this word fermentation to describe any microbial process or technology which is used to produce a microbial product by the mass culture of microorganisms. So when I grow microorganisms in a large mass and they produce a useful product for me, I call that process or that technology as fermentation technology. That is the industrial microbiologist's view. Okay. After having known this concept, let's also learn a little more about the industrial microbiological processes. These industrial microbiological processes, when do I set up an industry? When I find an organism that can convert a cheap raw material, that is a substrate, into a useful product which is of value, it is feasible for me to set up an industry whereby I will put this reaction on a large commercial scale. Now when I have to set up an industry, there are some of the prerequisites that I need to take care of. Now what are those prerequisites? We will talk about these prerequisites under 3 categories. The first one being the organism. I definitely need a microorganism because the first statement that we made was that this is a microbial process, microbial activity which converts a particular raw material into a useful product. Correct? So what we need to do is we need to know what the organism should be, how the organism should be, and what should be its characteristics that we have to talk about. Let's talk about the organism then. As is listed down here, the organism that we are going to use for an industrial production must be able to produce appreciable amounts of the product. That's the first property it should possess. If it is not capable of producing the product, I cannot set up my industry using this particular organism. So, it must be able to produce appreciable amounts of the product. It should have relatively stable characteristics. Most of these industrial strains which we use are mutant organisms, where I have made certain changes to their genetic DNA. Therefore, there are always chances that they will lose these characteristics. To take care of this, I have to choose an organism which has relatively stable characteristics, that is, the characteristics don't change. Thirdly, should have the ability to grow rapidly and vigorously. Grow rapidly and vigorously, the faster it grows, the faster it will produce the product for me. Therefore, definitely as an industrialist, it is of significance to me. And lastly, it should be non-pathogenic. You must have all heard about microorganisms being the cause of many diseases, okay, which are termed as pathogens. So, if the industrial strain is a pathogenic strain, it becomes very difficult for me to handle that strain. There are a lot of stringent controls which will be required. So, as far as possible, the microbial strain that I use for an industrial process should be non-pathogenic. So, these are the basic 4 characteristics that my organism should possess. Now, once a strain has been identified which follows or which is satisfying all these criteria that I talked about for the organism, my next step is the industrialist will have to undertake a research program. Now, what type of a research program will it undertake? It undertakes a research program to increase the capability of the microorganism. Increasing the capability of the microorganism will be with 2 respects. Either it increases the yield of the product, that could be the first characteristic property, or it increases the rate of formation of the product. That means I get more amount of product or I get product in a very short amount of time. Both of these features are very important for an industrialist. These things or these aims that I am opting for or I am looking out for are normally achieved by making use of customarily certain programs like improvement of medium and culture conditions, or I can select a new strain which gives me a better yield or a greater rate of formation of the product, or I could go for development of mutants. These have been the customary approaches that have been tried so far, okay? But more recently, with the advent of recombinant DNA technology, what is now tried is bioengineering of microorganisms, so that both of these purposes can be achieved. Now, let us look at how can I bioengineer a microorganism. The 3 steps, basic steps involved are, I need to have a source of donor genetic material, I need to then produce a hybrid DNA molecule, and lastly, I need to incorporate this hybrid DNA into a host cell. Let us see how can I do it. My source DNA is definitely going to be the property, the DNA fragment which codes for the property that I want to transfer into a bacteria. Let me explain this with an example. You must all have heard about human insulin. Okay? Human insulin production was very less and it was a very costly product in earlier days. But now, since microorganisms are capable of producing insulin, or we have rather engineered microorganisms to produce insulin, it has become much more cheaply available to us. So, this is the cell which possibly produces insulin, say for example. What I do is I take the DNA from this cell which has the genetic information to code for insulin. So, this becomes my source DNA. This becomes my source DNA. Now, the next step was production of hybrid DNA. For production of hybrid DNA, I need to have a vehicle or an agent who can carry a part of this DNA, because this entire DNA is not of use to me. I am interested only in that fragment of DNA which codes for insulin. So, I need to cut or tailor the DNA. Okay. Now, this tailored DNA fragment has to be inserted in a vector. A vector is an agent or a vehicle which is going to help me carry this gene from here into another organism or a bacteria. Okay. So, it is like a middleman which serves the purpose. What I do now is, source DNA is ready with me. The second step is production of hybrid DNA molecule. For production of hybrid DNA molecule, I need to make use of two enzymes, restriction endonucleases or ligases. Restriction endonucleases are molecular scissors. They cut the DNA. They cut the DNA, not only they cut the DNA, they cut the DNA at specific nucleotide sequences and generate well defined fragments of DNA. When I use a endonuclease, a restriction endonuclease for cutting the DNA, I find that the fragments which are obtained are identical. I am going to treat the source DNA as well as the vector DNA both with restriction enzymes, with the same restriction enzyme, so that it will generate identical fragments which are self-complementary. Just like you have a key for a lock, these are going to be self-complementary to each other. And now what will be my next step? Since I have fragment of the vector DNA, I have fragments of my source DNA. I need to combine the two to produce my hybrid DNA molecule. For this combination, for the hybrid, I make use of the enzyme ligase, DNA ligase, which brings about the ligation of the vector DNA. You can see that in the blue color, and the red DNA which is from the source. So, now what I generate is a hybrid DNA molecule. The question could be, what serves as the vector? The vector could be a bacteriophage DNA. Bacteriophages are viruses which infect bacterial cells. It could be a bacteriophage DNA, or I could also make use of a bacterial plasmid. Once I get this hybrid DNA molecule, I am left with one more step, and that is transferring this hybrid DNA molecule into a bacterial host cell. When I have to transfer it into a bacterial host cell, definitely I need to make the cell recipient to the DNA, because it is something external which I am trying to put into the cell. How do I make the cell recipient? There are two processes that can be done to insert the DNA. One is transformation and the other is transfection. When do I use transformation? When my vector DNA is a plasmid. When is the process called transfection? When my vector DNA is a bacteriophage DNA. Okay? The idea is the same. Transformation and transfection, both of them give me the same result. That means allow me to put the hybrid DNA into my host bacterial cell. The only difference is transformation process, the hybrid DNA, the vector molecule is plasmid, and in transfection it is bacteriophage DNA. This process, I said we will have to make the cells ready. The host cells will have to be readied to accept this hybrid DNA molecule. How do I make them ready? It's a very simple procedure to treat these microorganisms with calcium chloride. This makes the cells competent to receive this new hybrid DNA because it makes their membrane porous or permeable to DNA molecules. Once this is achieved, definitely I will get a variety of bacterial cells having different genotypes. Once I do transformation and transfection, I get many more such bacterial cells. From these bacterial cells, I have to choose the right one. Depending upon whether the characteristics that we mentioned earlier, it should be having stable characteristics, it should produce the product in larger amounts, it should be very growing fast and rapidly for a microbiological process to take place. So when I identify such an organism, what I then do is allow this bacteria to grow and multiply very rapidly and produce my required product. That was about one of the prerequisites of industrial microbiological process that we talked about, and that was the organism. The second prerequisite, as I mentioned, was a medium. Medium is the food that I am going to provide to these organisms for growth. This medium that I am using should have definitely the substrate which the microorganism is going to act upon and convert it into a useful product. Now this substrate which I am employing should definitely be cheaply available, because I have to look at it from an industrialist point of view. It should be cheaply available, as well as it should be available in large quantities. Okay. Once this is achieved, then my medium composition becomes much more easier to develop. The medium could also be the waste of other manufacturing industries. For example, from the dairy industry we get a waste which is called as whey, which is highly nutritive and could be a problem for environment. So if I can use this highly nutritive waste of another industry as the raw material or the medium for producing a new useful product, I am saving on to the cost. Okay. So the medium could definitely be either a completely new material, or it could be a waste from already running microbiological industries. Thirdly, we come on to the product. Microorganisms are made to convert this substrate into the product in large tanks, which are termed as, these fermenters have a capacity not less than about 50,000 gallons. So you can imagine that this much is the volume that I need to take care of medium, and it is a very rich medium which is provided for the growth of the microorganisms. Now when the fermentation takes place in this fermenter, when substrate is getting converted into the product which is formed is going to be present in a heterogeneous mixture. Why do we say it's present in a heterogeneous mixture? For the simple reason that this medium contains microbial cells, it contains unused media components, it contains the used media components, it contains waste products which we are not looking out for. We are looking out for a particular product that is of value to us, but there could be many more products formed by the microbial activity. So now what am I left with to do? I just cannot take the medium out of the fermenter and sell it as my product. What I need to now do is have economical, easy to operate recovery processes, which are going to allow me to screen off the product from the fermentation media, and I can then subject it to further purification. Now this is very important, because you can imagine if you were supposed to have an antibiotic for treatment of some bacterial infection, and if that antibiotic is contaminated by microorganisms, it can cause diseases. In the person who is using the antibiotic to cure himself, it can lead to untoward effects. Okay? So I need to purify the compound that is being formed. So the prerequisites for an industrial microbiological processes we have talked about with respect to three: organism, medium and the product. Okay. Let's now go on to know a little more about the fermentation technology with an example. This is the diagrammatic or a schematic representation of the production of penicillin. In the simplest of the terms, it's much more complex, but in the simplest terms, what I have put up is a diagram which shows the manufacture of penicillin. As you see, it happens in 4 steps. Step A, that is preparation of medium. Step B, which is preparation of inoculum. Step C, that is the fermentation. And step D, which is the recovery of penicillin. Now, what happens in step A? Media ingredients like corn steep liquor, salts, lactose and many more constituents that are required by the cell for its growth are mixed together in this mixing tank. This medium is then sterilized. Sterilized medium is then cooled and then pumped into the fermenter vessel. Okay. So, the medium is now ready for fermentation. Step B is preparation of inoculum. You can see here the tube. This tube or this slant has the culture Penicillium chrysogenum, which produces penicillin for us. This is the organism which actually led all these microbiological industries to flourish. This was the first product that was obtained. This Penicillium chrysogenum, definitely I store it in small tubes in the laboratory. When I want to put it into the fermenter vessel, what I do is I develop the culture. This is called as preparation of inoculum. So, from the tube, I put it onto bran where the spores multiply, and these spores are then transferred into a seed tank. From the seed tank, enough of volume of the inoculum is built up, and this is then added into the fermenter. Now, step A completed, step B completed. Coming on to step C, which is the actual proper fermentation that happens. What happens here in the fermentation? You've added the medium, you've added the culture. The minute you provide growth nutrients to the microorganisms, they are going to convert the substrate into the product. What is the product that we are looking out for? We are looking out for penicillin production. So, this product is going to be starting to be formed in the fermenter, but it just does not form immediately. It takes incubation time, which may be from days to even months. The culture is allowed to grow and develop, and the penicillin is now produced. Once this fermentation is complete, as I said, we need to have a recovery and purification process. This is done here. First step, filtration, whereby the mold, that is Penicillium chrysogenum, which is a fungi. Fungi are also known as molds. These are separated out by filtration, followed by recovery processes, whereby I carry out precipitation, I also carry out solvent extraction and get this penicillin in a more purified form. So, what we have learnt about is, for the basic process of microbial industrial process, there could be different sections. Basically, there are 3 sections. First one being upstream processing, second one being proper fermentation, and the third one being downstream processing. Now, I would like to know, which do you think come under upstream processing? A, B, C and D. Of these, which are the parts that come in upstream processing? Correct, A and B. As the word suggests, this is upstream processing. So, this is the preparatory stage that is happening. So, before the fermentation, whatever happens, they come under upstream processes. Okay. Once the upstream processes are completely carried out, then I go on to the main fermentation. This main fermentation that happens here is the second part, that's called as fermentation proper. Then going on to the third part, and that is downstream processing, which will involve all my recovery processes like filtration, centrifugation, sedimentation. I can also go for analytical methods like chromatography, etc. Okay. So, this is a basic fermentation cycle that we are talked about, looking at one of the products, and that is penicillin. Let us now come on to the next part, which we are going to talk about in a little details, and that is inoculum development. What is inoculum development? What do you call as an inoculum? say we talk about the same example, penicillin. This is the organism Penicillium chrysogenum, which produces penicillin for me. When this organism is capable of producing penicillin to me, it is called as inoculum. Okay? But why do I need to develop the inoculum? The word says inoculum development. Because as I mentioned earlier, the fermenter capacity is of about 50,000 gallons, okay, or more than this. If I just inoculate one tube of a culture, it will take enormous time to complete the fermentation run. So when I have to carry out a fermentation on an industrial scale, I cannot allow for the loss of so much of time. And therefore, what I need to do is have a large volume of the inoculum ready with me to be inoculated into the production fermenter. So how do I then define inoculum development? The handling of the inoculum, that is the culture, from the preserved stock, from the time that it is transferred from the preserved stock until the time that it reaches or it is inoculated in its final location, that is the fermenter, all the processes that are carried out, they come under inoculum development. And my primary purpose of inoculum development is to make available large amount of the inoculum at the required time, such in the right morphological state, so that as soon as I inoculate it into the fermenter, it allows for the product formation to take place. And I need to reduce my lag phase. If you remember the growth cycle that I talked about, this phase is a wastage of my time because no product is being formed here. So as an industrialist, I need to make it as less as possible. When can I make it as less as possible? When I have a large amount of inoculum ready with me and I put it into the production fermenter. Let's look at some of the criteria that the culture should possess to be used in the fermentation process. Firstly, the inoculum must be in a healthy active state, thus minimizing the length of the lag phase, which is the unproductive phase. Secondly, it should be available in sufficiently large amounts or volumes to provide an inoculum of optimum size. It must be in a suitable morphological form, whether I want it in a sporulating form, whether I want it in a vegetative form, I should be getting it in that form. Next, it should be free of contamination. Contamination could be from a variety of organisms. Contamination could be from air. Contamination is anything, any other microorganism other than Penicillium chrysogenum being present in my fermenter vessel. That is called as contamination. Any living organism present in the fermenter medium other than Penicillium chrysogenum if I am carrying out production of penicillin, that is termed as contamination. So it should be free, completely free of contamination, and it must retain its product forming capabilities. It should not lose the product forming capability. Otherwise, the minute I put it in the industrial fermenter, it is a waste of my medium constituents, it is waste of lot of money which goes into it. Okay? So these are the characteristics that need to be taken care of. Once I find such a strain which is satisfying all of these criteria, definitely I can use it, or I can allow for inoculum development now to take place. Let's look at the principles and various aspects of inoculum development. Firstly, pertaining to the inoculum size. The inoculum size ideally should be between 3 to 10% of the volume of the production medium. It should be between 3 to 10% of the production medium. Okay. So if production medium is 100 ml, I can do with say 3 ml to about 10 ml of the inoculum. But as I mentioned earlier, it goes to about 50,000 liters and much more than that. So you can imagine how much of amount of inoculum will I require for my process to begin and go to completion. Okay. Therefore, now starting from one tube of culture that I have as my preserved stock culture, I need to develop this much of inoculum. Okay. Now this cannot happen in a single step. So what I do is I break down this process into several steps. When I break down this entire process into several steps, at each step I increase the volume of the medium. So from the tube I will put it into a 100 ml flask. From a 100 ml flask I will put it in a 1 liter flask. From a 1 liter flask to a 5 liter fermenter, and so on, depending upon how much of the volume of the fermenter do I need. How much of the volume of the inoculum is required to be inoculated into the production fermenter. Therefore, this process, this sequence of steps wherein progressively I am developing my inoculum, that is I am increasing the volume of my inoculum, all of it comes under the process of inoculum development. Okay. Now, when I carry out these steps, when I am transferring from one step to the other, to the other, to the other, the risk of contamination is very high. There is also a risk of strain degeneration. Therefore, stringent quality control procedures are required during the development of inoculum. And all of these needs to be done under completely sterile conditions. Once this is achieved, one more factor that I need to consider while I am talking of the volume of inoculum to be developed, and that is of the cost. Economic consideration also has to be given, because I said 3 to 10%. For making 10% of the volume, finally I will need another fermenter vessel which will be of the capacity 10% of my production fermenter. Now when I am going to build another such fermenter, definitely it is a financial investment which I need to make, and that needs to, the use of this fermenter has to be justified. Okay. So these are the two factors that I need to consider. Next is the inoculum medium and incubation conditions. When I use an inoculum medium for inoculum development, it should be such that it allows for large amount of biomass to be produced, but less or absolutely no product to be formed. Okay. So if I am using a inoculum development medium for penicillin production, it should be such that it will allow for the rapid growth or rapid spore formation of Penicillium chrysogenum, but will not produce penicillin. Okay. Another consideration that I need to make with respect to this point is that when I am using the media, it should be such that in case if my production medium involves any enzymatic adaptation, then that inducer molecule should be a part of my inoculum medium also. Thirdly, some of the processes, some of the fermentations, they require the separation of the cells or the spores. Ideally, I can just directly produce it in liquid media and then transfer it into my main fermenter vessel. Sometimes what is required is the cells be separated out, the biomass be collected, separated out, and then added to the production fermenter. This is normally in the case when I need, instead of 3 to 10%, I need an inoculation of up to 20%. That is the time when I separate out the cells and then add it directly to the fermentation vessel. This allows me to take care of the dilution problems which can otherwise take place. Also alterations of pH which can happen. So I need to take care of the cells alone. I remove all the liquid part or the media components and constituents and only concentrate on the cells. The third is the technique of transferring the microbial growth from the stock cultures to the liquid inoculum media and initiating the growth in it. Say for example, if there are bacterial cells, I can just directly inoculate, they will start utilizing the media and will build up the biomass. However, if there are certain bacterial spores, say for example belonging to the Clostridium species, they need to be given heat shocking effect for germination to be induced. But if I cannot generalize the term, because fungal spores are sensitive to heat, so I cannot use heat shocking for them. So these all considerations have to go in when I am doing the transfer of microbial culture from the stock culture to my final production fermenter. Also, in some cases like non-sporulating fungi etc., I need fragmented hyphae to be inoculated. So how do I fragment the hyphae? I scrape the growth of these fungi or actinomycetes from the agar surfaces with the help of a sterile nichrome wire or making use of other means, and I scrape it vigorously. So this fragments the hyphae and this is then inoculated for inoculum buildup. Coming on to the next principle is the high frequency of back mutations which can occur. This can definitely be prevented by the right choice of media and incubation conditions that permit only the desired strain to develop in the medium. The last principle, that is contamination. When I talk of contamination, if it is a fungal production, I can easily detect heavy bacterial contamination because it will produce uniform turbidity. However, I cannot rely on that. What I normally do is I carry out microscopic observations, determine if there is any contaminant. I also sample the material obtained from the inoculum build-up process on plates, on agar media to check for any contamination, and this has to be done on a regular basis. After knowing all this principle, let's just revise what we've talked about with respect to inoculum development. I said it is a sequence of steps before it comes into the final fermenter vessel. See here, the culture of producer organism on an agar slant is present. One loopful, a loopful is just as small as a water droplet on your bench. That much is inoculated in 30 ml. That's the first step that happens. Once this culture grows, it is then incubated for growth. Once it has grown completely, 30 ml of this seed culture is put into a 300 ml of second seed medium. The volume increases. Loopful, one loopful of spore inoculated here. Now I am inoculating 30 ml in the next flask. Again kept for incubation. Now 1.5 liters of this is added into the 15 liters of third seed medium. Okay, again increase in volume. From here, allow again for its growth to take place. This 1.5 liters from here is added into 60 liters of the fourth seed medium to give me so much of volume of the inoculum alone. Finally, it is allowed to grow, and from this last seed culture, I inoculate 60 liters of the fourth seed culture into my production fermenter, and then allow for incubation, where it will now convert the raw material into the required product. Okay. So this production or this inoculum development is for the development of Saccharomyces, which is produced by the microorganism Micromonospora species. Okay. So that's about your inoculum development. Just to recapitulate what we have done, we first talked about the importance of microorganisms in industry. We then listed down the major classes of microbial products. We talked about the concept of fermentation technology. Then we took an example of an industrial microbiological process and what are the three things that we need to look at it. That was the organism, the medium and the product. Then we learned two more terms, that is upstream processing and downstream processing. Later on, we talked about inoculum development as a whole. We talked about the criteria that the culture should possess. Later on, we talked about the various principles which influence inoculum development. Okay. So with that, we have basically covered what do we understand by inoculum development in fermentation technology. Thank you, all of you.